The next one is the in vivo. Comparing to all other threes, this is one of the best preclinical methods, because the same human studies, directly we are giving the human study, the as it is animals we are using for the trial here. The in vivo is nothing but inside the living things or inside the animals, living organisms. This is the in vivo, inside the living organisms. The conducted on whole living organisms to study assess systematic drug actions. It means that we are conducting a study here on the whole living organisms to assess a systemic drug action, whether drug is acting or not, that we are analyzing on the whole living organism as it is in the living organism. This is the principle in the in vivo method. The next one, example. Example is here the amlaki. You can take Amblica officinalis amlaki here. How we are using? Okay, you can take here the rat. In the animal studies, we have to separate, take the rat. First of all, what we have to do is, if you want to check the hepatotoxicity, the liver, whether the drug is toxic to the liver, the hepatotoxicity is there or not, whether the drug is affecting against the hepatotoxic, that we are checking. Here the example is, take one rat. In that rat, we have to give this amlaki or Amblica officinalis extract, the fruit extracts that we are giving, and after that we have to give the CCLH. It means that carbon tetrachloride. Carbon tetrachloride is one of the toxic drug, that is the liver toxicity. It will produce the liver toxicity. If we are giving the carbon tetrachloride as well as the amlaki, whether this drug is protect the liver or not, that we are analyzing. Like this we are doing the experiment. Okay, this is the one of the example in the in vivo toxicity. If in their liver function test, suppose normal or liver looks normal in their size shapes, it means the drug is hepatotoxic. If it is not safe, then it is considered that CCl4 carbon tetrachloride is the toxic, but it is not preventing the liver. Like that we can analyze here using this method. This is about the in vivo method example. Next one is the applications. Application is the pharmacokinetic study. By using this method, we can analyze the pharmacokinetic study, that is the ADME, how the drug is absorbed, how the drug is distributed in our body, how the drug is metabolized in our body, as well as how the drug is excreted. All these pharmacokinetic study we can analyze using this in vivo methods. Along with the next toxicology also, whether drug is toxicity it is there or not, that also we can analyzing by using this in vivo methods. Next is the screening whether the drug is safe or not, that also we are analyzing. Next one is the pharmacodynamic evolution. Pharmacodynamics means the drug is effective or not, that also we are analyzing using this in vivo methods. The next is advantages. Why we are using this in vivo method in the preclinical study? Advantage is provide complete biological response. Okay, the whole animal we are using here, the whether complete biological response we are analyzing in this study. That's why it is one of the benefit of using this in vivo method. The next is the lastly limitations. What are the limitations of in vivo methods? In vivo method, it is a costly, because we have to collect the rats and we have to go to the animal house. That's why it is costly. Ethical concerns, because for that we have to take the permission for the animal ethical committee. That's why also it is ethical concerns are there. And species difference. Lastly, species difference, because we cannot tell that the specific animals in that this drug is safe or effective. The same it will act safe in the human beings or effective as it is, we can't tell. That's why this is one of the limitations, is the specific species difference also we can analyze. This is the limitation, it is there in the preclinical study in the in vivo methods. These are the 4 methods we are using in the preclinical trial before giving to the human beings. Hope you understood the today's topic. If you understood, please share to your friends, like, share. Thank you.